7th grade, this is the information for Thursday, November 12th, Friday the 13th of November, as well as Monday, November 16th. On Thursday, we will go over Wednesday. Today's lesson is cell types and Gizmo, and we will preview Thursday and Friday. So on Wednesday, you learned about the microscope. You were supposed to watch the video and look at the diagram and then do the practice assignment. If you have not done these things yet, please pause this video and do those 3 things right now. Again, the video, the diagram, and then the practice. On Thursday, students will be doing a cell types gizmo where we are using a compound light microscope to understand that all living things are made up of cells, and then we're going to compare and contrast some different cells. Again, if you haven't done this on your own, this is something you can go through by yourself. Please pause this video, complete the cell types gizmo, and then come back and check your work. If you're rejoining us, that means you have completed this gizmo. The first thing you were supposed to do is look at some prior knowledge questions, thinking about hopefully those things that we said the other day about what makes something alive. They use energy, they respond to the environment, they grow, they reproduce. And then the second part is, I can't see, humans, plants, and mushrooms are alive. What do they have in common? They have those characteristics in common. The warm-up here is making sure we're looking at the Elodea leaf here correctly. So when you did that, you saw rows of shapes that look like little rectangles, kind of like bricks. Then you needed to look a little closer and you saw some shapes inside of those rectangles. The rectangles are cells, and then the parts inside of cells we call organelles. Here we are looking at the compound microscope. And so you were supposed to become familiar with the different parts and match the definition or description to the diagram. Here are the answers to those questions. Oops, I lost the answer somewhere. Oh, this is a preview, that's why. Here are the things that you're going to do. This is from my class. So here are the correct answers. Again, if you've done this, the stage is B, the eyepiece is A, the coarse knob is C, the smaller knob is the fine knob, the objective lenses are E, and then the slide is F. Here, it's easier to use the 40 magnification on coarse, the 400 on fine, and on the human skin cell, you're able to see the nucleus, the cytoplasm, and the cell membrane. Then you are supposed to look at the mouse skin, a fly muscle, a maple leaf, the elodea, and fungus, and then look at the different organelles or parts of a cell that are visible. So here we see the different organelles. All the samples contain cells. All of the cells have a nucleus, and we call those eukaryotic cells. Eukaryotic cells. We didn't have to measure the E. coli, but we saw some things present in it: a cell wall, a cell membrane, a cytoplasm, flagellum, pilus, and a nucleoid. It's missing a nucleus, and so we call these prokaryotic cells because they do not have a membrane-bound nucleus. Then you looked at sand or silt, and it is not alive. It doesn't have cells. It doesn't grow, reproduce, and do all of those things that living things need to be able to do. Then here we're looking at the different features of those 4 cells that we have to be familiar with: the nerve, the skin, the muscle, the blood. They have nuclei, all of them except the blood cell. They have cell membranes, they have cytoplasms, and then some special structures that were noticed. Then you were supposed to draw or take a screenshot of what those look like. So here from the gizmo is what the neuron, the skin, the muscle, and the blood look like. And again, what is the function? Neurons transmit and receive signals. Skin is for protection. Muscles contract and relax. And red blood cells, make sure we're focusing on just the red blood cells there, they carry gases. Extend your thinking. You can pause and look in that. Friday's work is watching the video, reading about what's found inside a cell, and then completing assignment 9. And then Monday's a continuation of that, looking at plant cells, being able to label diagrams, and then being able to identify the functions of the organelle.